So today is meant to be somewhat informal. I'm going to bring up some topics of materials design. I'm going to bring up some topics in tissue engineering, but feel free to stop and ask questions about things as you will. I also am going to go back and forth between a couple of different talks because I realize I want to say say a couple of things that I didn't put in the formal presentation for you guys. And one of these is that a major interest, I know you've heard a lot of things in the past couple of weeks about development and mechanical forces and so on. One of the things that fascinated me as I got into the field of tissue engineering, which I came into from chemical engineering and got interested in biomechanics and mechanical forces in large part from interacting with Roger through our educational programs. But one of the things that really inspired me when I started in the field many years ago and was thinking about how do you control cells with molecules on the surface was that there was fascination with putting RGD on. Everybody know RGD, the cell adhesion peptide, okay, that came out of fibronectin. It's a loop in fibronectin and it allows cell adhesion. Well, you know, a lot of people in biomaterials were putting RGD on surfaces, but when you go back and look at nature, in fact, RGD motifs, as well as other adhesion motifs, were extremely highly organized in the extracellular environment. And in particular, different facets of physiological processes would have matrices with different degrees of organization. And in particular, because I was in tissue engineering, which has many, if you want to grow a tissue, there's many similarities to both development and wound healing. There was a molecule that was particularly fascinating called tenascin C, and there's a picture of it shown here. It's a hexabrachian, 6 arms, and what you have in here are EGF-like repeats. So the EGF motif, epidermal growth factor motif, I'll show you a picture later, has 6 disulfide bonds, 3 disulfide bonds, very stable molecule, and the motifs appear in lots of matrix proteins. And it's unclear whether they have any signaling functions or not. But there's 13 EGF-like repeats, and then several different adhesion domains, including RGD loops that interact with integrins alpha-5 beta-1 and alpha-9 beta-1. So the structure of this molecule was fascinating because it suggested that maybe one role of matrix is to organize receptors together on the surface. Okay, so that part of the way that cells are responding to their environment, it's not just stiffness, but it's also this nanoscale organization. Maybe the ligands in the environment are telling the cells where to go. Or telling receptors to cluster together or not cluster together. So maybe tenascin was meant to bring EGF receptor and integrins together in a very close cluster that would have special signaling properties. And this is important because tenascin is found very prominently during the early stages of development. Tenascin C, there's several forms. It's also found in wound healing. So when you have a skin wound at the front where you have the granulation tissue meeting normal tissue, you see tenascin. And it's found in tumors. So a lot of cancers overexpress tenascin. And it's also found in the bone marrow in the hematopoietic niche. So it's a very interesting molecule having to do with proliferation, stem cells, and so on. And so as we were thinking about how to take inspiration from natural molecules, thinking about the biophysics of this, we said, gee, this approach then of putting adhesion ligands on a surface to engender cell adhesion, maybe we should be thinking not just about is it RGD or a collagen domain, but maybe the biophysical presentation is important. And I don't have a slide to show this specifically, but you can imagine if a cell has, say, 100,000 integrins, and it's going to attach to a surface, if the ligands are distributed randomly, okay, the integrins are going to bind wherever. Some may be clustered together, and some may not. And there was a lot of literature at the time from the biology community suggesting that integrins actually had to cluster together in order to link up to the cytoskeleton and signal. So that one integrin bound to a ligand wouldn't nucleate talin and vinculin and all these things you think of in focal adhesions. You actually needed several of them together. Okay, and now we know it may be not only just the integrins, but syndicans, maybe growth factor receptors. But the general idea is several things in one place. So one could reason if you have a stochastic elastic distribution of ligands on the surface. You're going to get integrins bound everywhere, and you may use some of them up in unproductive interactions because they're just kind of sitting out there by themselves. So we did a fairly straightforward experiment where we took a molecule of polyethylene oxide that's a star shape. It had lots of arms coming out of a central core. And we put it on to surfaces, and we varied the average ligand density with a RGD, very simple low affinity RGD ligand. So we varied the average ligand density. But we presented it either randomly or in clusters that were small or clusters that were larger. And we examined facets of the cell biology, including migration of these cells. So this is just showing speed of cells as they're migrating on these surfaces. And what you can see is, so for example, take this density, is that when you cluster the ligand, you get a much enhanced speed. And this was also, you get enhanced focal adhesion formation, so on. Larger clusters lead to an even larger increase with the same ligand density. Now, you don't get this biphasic behavior. Have you seen the biphasic migration curve so far? People not seen it? Okay, so the cell migration process involves a cycle. So you have cells adhering. So, and is the movie going to play? Can I play the movie? Maybe it's not in there. Okay, the movie's not going to play. But you can see these cells. They extend a lamellipod, so you get an extension of the cytoskeleton out. And if they're moving on a 2D surface, as they are here, and then they form adhesions at the front. Okay? Then by mechanisms that are still not completely understood, they generate an asymmetry between the front of the cell and the rear of the cell, and then they contract. And since they're more adherent at the front, they scoot forward. Okay? So you contract, you're adhering at the front, you're not adhering so strong at the back, you can move forward. Okay, so there's a cycle of this, and as you can imagine, the speed that the cell moves is going to depend on the ratio of this ability to contract and how strongly it's adhered. Okay, now imagine you're walking across ice. It's very hard to get traction, right? You don't move very fast because so it's like a cell on a poorly adhesive surface. So you, so Doug, I'm fine if you want to. I think I'm going to be okay. He's worried that I'm going to faint or something. So it's like a cell. So if a cell is on a substrate that doesn't have much ligand, it has a hard time getting traction. So it extends its lamellipod out, it adheres, but when it contracts, it pulls up from the front and the back, so it can't really move. Okay, but now if you increase the amount of adhesion ligand, now the cell can balance this contraction and adhesion, and it can move forward. Okay, but now as you keep increasing it, more and more and more, suddenly it gets like you're on flypaper. Now you contract, but you can't move because you're still stuck in the back. Okay, does that make sense? All right, so that's a classic model. It was actually developed by Doug Laufenberger's group, and he and his collaborator Rick Horowitz wrote a seminal paper in Cell many years ago. I can't remember what year it was published, but it was before he was at MIT, so it was more than 15 years ago, and laying out that this kind of biophysical model of cell migration could predict how changes in integrin expression, integrin affinity, ligand density, and so on would influence cell adhesion in 2 dimensions. And this model has been held up over and over and over again by experimental measurements from labs all over the world, including like Claire Waterman Storr. Okay, so coming back to biomaterials, because that's really what we want to focus on, what we see here is we're not capturing that biphasic behavior with our ligands. And you can imagine the student who did this, we pressed her to get more data here, but she said, no, we don't actually need that data because the true independent variable is not how much ligand is there, it's how strongly the cells are adherent, going back to that model. So in fact, if we go back and take these data and add to it other data saying how strongly the cells are adhering to these substrates and replot it, we find that all these data here are collapsing onto one curve where you've got the speed as a function of cell adhesion strength, right? And so the problem with the synthetic ligands is they follow the curve, same curve as fibronectin, they just don't go up as far, and they're limited by the affinity of the ligand that we use. So we use a very low ligand, so you simply can't get enough bond strength to move over into this regime. Okay, now you see there's also two curves here. There's one that says plus EGF, epidermal growth factor, and one minus EGF. EGF is a growth factor you add in soluble form, and it stimulates a lot of processes that influence migration. It stimulates contraction of the cytoskeleton. It stimulates activity of a molecule calpain that cleaves integrin cytoskeletal bonds. So there's multiple ways that epidermal growth factor can stimulate migration, so that if the cells can contract harder, then they can also move faster. So EGF can stimulate migration. But the nice thing is all the data fell onto these two curves, one with EGF and one without EGF. And if we'd gone in and tried to replot it as a function of the ratio of contraction to adhesion, they would have all gone on one curve. Okay, but measuring the contraction for these was too hard. Okay, so I mentioned in the last slide epidermal growth factor. I'm going to come back to that in the context of mesenchymal stem cells because it's a molecule and receptor system that's very important in development. So most of you know what insulin is, right? You know, insulin. Epidermal growth factor is very similar to insulin in size and somewhat in structure, not exactly. This is a picture of it, 3 disulfide bonds, very compact molecule, about 6,000 molecular weight. It's made and it binds to a receptor on the cell surface that's very similar to the insulin receptor. It's a tyrosine kinase type receptor. But what you see here is epidermal growth factor, like many growth factors, is made as a transmembrane precursor that gets cleaved by proteases at the cell surface. And in fact, the EGF receptor family, or EGF, was the very first growth factor ever discovered. And it was discovered in the 1960s. There was a Nobel Prize that was awarded for this. A bizarre experiment. They smeared mouse submaxillary gland secretions onto incisors of newborn mice, and their teeth grew faster. That's how they discovered it. I don't know how people can do these experiments. But so it plays a big role in epidermal development. But it turns out it's a prototypical molecule of many similar molecules that can bind to the epidermal growth factor receptor. So what's shown here are epidermal growth factor, which is made as this huge molecule, and then it's processed to just give that little motif shown there. Amphoregulin, beta-cellulin, epiregulin, epigen, heparin binding, EGF, and TGF-alpha all bind to the EGF receptor. Now, what I forgot to mention on the intro slide with tenacin is that with our collaborator Alan Wells, who's now at Pittsburgh, we showed that one of the EGF-like repeats in tenacin also binds to the EGF receptor and can stimulate migration through what we call a matricline fashion. So it may be so. How many of you have used EGF in an experiment or used any kind of growth factor in a cell culture experiment? You did some cell culture experiments this week, right? Yeah. Did you put PDGF or VEGF or anything in the culture medium? Anybody put anything in a culture medium? Yeah. What did you put in? You don't remember. What did they grow, Roger?
They grew myocytes and they grew neurons.
Okay. So did you put nerve growth factor? Do you do NGF for that, or were you just given like a mysterious? You put retinoic. Okay, retinoic acid is a little bit.
Oh, and GDNF.
Okay, glial-derived GDNF, glial-derived neurotrophic factor. Okay, so that was the soluble molecule, right? It was in the medium. Okay, so you think of, most people think of EGF as this reagent you get from Sigma and, pipette into your cell culture media. But in fact, if you look at these are all made as membrane-bound precursors. Okay, they're not secreted. Insulin is secreted. And so here's where a big difference is. The insulin secreted, these are made as membrane-bound precursors that sit on the cell membrane until something cleaves them off. And many growth factors are like that. I don't know about GDNF, but a large number of growth factors are like this. So this depicts now going into when you cleave the ligand, so the ligand's sitting on the surface here, it can act in a juxtacrine fashion, meaning it can stimulate a cell that's next to it. Now, not all of the ligands can do that, but many of them, HBEGF, can act in juxtacrine. And then it can be cleaved by proteases that exist on the cell surface. So there are a number of different proteases in an area of very intense interest to understand possible cancer targets. How do you inhibit these things being cleaved when they shouldn't be? And then it can come off as a soluble ligand and act either in an autocrine fashion, stimulate the same cell, or in a paracrine or endocrine fashion and stimulate either a neighboring cell or even go into the body fluids and stimulate something far away. So EGF in particular helps keep your GI tract healthy. So your GI tract is bathed in EGF, and because your intestine cells are being constantly bombarded with all kind of toxins and stuff, so they need to turn over a lot. Okay, so that's just a little intro. Finally, the receptor for epidermal growth factor, and we will get on to mesenchymal stem cells. I just want to do a little bit of biology, is shown here, and it's a typical tyrosine kinase receptor, which has an extracellular domain that binds to a ligand, EGF or ampiregulin or so on, and an intracellular kinase, a transmembrane domain, and then an intracellular kinase domain that puts phosphates on, phosphorylates other proteins, and with a regulatory tail that helps control activity of the kinase domain. So EGF receptor, usually, again, this is hotly debated about all the aspects and how it's activated, but there's evidence that exists in a tethered conformation, and then when it binds to a ligand, you get it straightening up, exposing a dimerization arm, and then two receptors come together, and then they transphosphorylate each other. Okay, so this is a paradigm. This need to interact with another receptor in order to be activated is a common theme in the EGF receptor family. Okay, so that's a very important thing. So that's some basics on the EGF receptor. And what I really want to do is give you, so that's some nice cell biology, and a lot of my lab, we focus on manipulating EGF receptor family for tissue engineering and actually also for, more recently, chemotherapeutic agents. So what I want to mainly talk about this morning is strategies to repair connective tissues in the clinic. And this derives from about a 10-year collaboration with George Mushler, who's an orthopedic surgeon at the Cleveland Clinic. And George started his career as a surgeon who was really the go-to person for removing large tumors in bones. So this is one of his early patients who has an osteosarcoma, which is a bone tumor, and you can see it here. And so what he became expert in doing is removing this affected area and then replacing the area with a piece of cadaver bone. So that is the gold standard. And there are options. You could have the leg amputated, and that's what they used to do like 50 years ago or 30 years ago. But in recent times, there's been a lot of effort put into salvaging limbs when there's a huge defect that requires excision of a damaged area. And so the standard is to just take a piece of cadaver bone and bolt it in and then let the patient heal. Of course, you think that heals very well? No. That bone takes a long time to turn over, and there's all kind of issues with immunogenicity, well, more concern about transmittal of disease, and so on. So this sparked, and later in George's career, in Dr. Mushler's career, he still does that, but he also treats a lot of orthopedic injuries that you would call a large segmental defect. So a large segmental defect is you have, like, you break your arm, and for whatever reason it doesn't get fixed properly, and it doesn't ever heal. Okay, the fracture doesn't heal, and then if the fracture doesn't heal, what happens is the edges start to erode away. Okay, and so then you get this gap that's never going to heal. There's something in bone called a critical size defect. In fact, there probably is in most tissues. So people know what a critical size defect is? Can you guess? Yeah, what do you think it is? If you break your bone and there's just a little gap, it'll heal by itself, right? Okay, now if it's a bigger gap, it may still heal, but then you get to a critical size where it's never going to bridge the gap. That's called a critical size defect. And you probably have that in your skin. If you get a paper cut, it heals, right? But if you kind of scrape really far down, you'll get a scar, right? It repairs, but it doesn't heal. right? It doesn't regenerate, it repairs. So there's some balance probably for every organ and tissue between this regeneration and repair, where you get a small wound, it'll fill in, you get a large wound, and it repairs with scar tissue but doesn't really regenerate. Okay, so in bone, in people, that's about 1 centimeter. And so what the other approach, so a sarcoma, you have this huge thing, you take it out, but in a, like say you break your arm and it doesn't heal, the surgeon has to go in and you have to put something into that defect to get it to heal. Okay, because you can get it to heal, it's just it needs some help. And it needs some help with two things. It needs some kind of scaffold for cells to grow on, and it also needs some source of bone-forming cells. Okay, so if you have a large critical defect, you want to put something in there that gives you a source of cells that will form bone and a scaffold for them to grow on. And you absolutely need those two things. And then a way to recruit blood vessels in and so on. Okay, so again, clinically, what do people do? They will take a harvest of the hip bone. So anybody know anyone who's had that procedure? You go in and you have a critical size defect, and the surgeon will not only open up and clean out your wound, but they go in and they have this thing that looks like a melon baller, and they take the dense cortical bone off, and they go in the trabecular bone, the really spongy bone in the inside of the hip, and they scoop out little melon balls of bone, and then they put it in the defect. Okay, so that's what they'll do, and that's called an autograft. And the autograft has marrow in it, so it's got cells that form bone. Okay, so that's what they do. Okay, so interestingly, what ends up with the patient, that the site that they harvest the bone from hurts for a really long time. There could be all kind of complications from harvest of the autograft. So this procedure, while it will work and restore function in a defect like a critical size defect, it's not preferred in the long run. If you could get around doing that, you'd like to get around doing it because it would really improve the outcomes for lots of patients. So in the community, including work that he and I do together, there's tremendous interest in trying to design graft strategies that would include osteogenic cells and scaffolds, but not necessarily require the actual harvest of bone. So can we design a synthetic scaffold where we control something about the scaffold that helps the tissue grow in, but at the same time we also control, and here now you can see the cells migrating. These are actually migrating on a tethered, an EGF, tethered EGF surface. But that we can also design the surface so that we enhance the function of the bone cells. Okay, so as a first step, what George Mushler did is show, okay, so we know, and I'll show you some slides of how we know this. Oh, wait, actually, let me go back to my other talk. I meant to go back to my other talk. Hold on. I apologize. They gave me like these massive drugs for that bee sting, and I'm not quite awake. I meant to just show that tenacin and then go back to this other talk that has some more tutorial stuff. So we'll go back to it now. Okay. Okay. So I want to come back before we'll come back to the bone tissue engineering. So let's take a step back and say we want to talk about the osteogenic cells. So it's just a little bit of a disjoint, but not too much. Okay, so a lot of what we know about marrow in terms of the connective tissue progenitors that are in marrow comes from some amazing experiment done by this guy in one of the Russian provinces, Friedenstein, who started in the 1960s to observe what happened when you take marrow and just plate it in culture on like a culture dish. And he used animals, he used hamsters and rabbits and maybe later mice, I can't remember. But he would take the marrow and he would take all of the mononuclear cells, so all the nucleated cells, and put them in a serum-containing medium and then watch what happened after a couple of weeks. And you would always see, when you did this at a low plating density, you'd always see these colonies of fibroblastic-like cells grow out, okay, in the presence of serum. Okay, now, think about it. You've got all of marrow there, and now the only thing you see after a couple of weeks are these fibroblast colonies. You're changing the medium. Why wouldn't you see any of the other, what other cells are in marrow if you take the marrow out and put it in culture? What other cells are present in marrow? I know it's Friday, nobody wants a quiz, but surely someone knows. What color is marrow? Marrow is very red, right? That's a hint. Red blood cells, and where do red blood cells come from? What's a word that means creation of red blood cells? Hematopoietic system. So erythropoiesis is specifically red cells. Hematopoiesis are the cells in blood. So the white cells and the red cells come from a process called hematopoiesis. And there are stem cells and more lineage-committed cells for hematopoiesis in the bone marrow, right? So we think of bone marrow, we mostly think of hematopoiesis because you get bone marrow transplants. You're getting the function that you're after is this hematopoietic function. So when you plate marrow, what are most of the cells stromal cells, or are they hematopoietic cells? Anybody got any idea? No? They're mostly hematopoietic cells. So you've got, if you look at your bone marrow, it's churning out, because your blood cells turn over. You know, your red cells live an average of 120 days. Your white cells, some of them only live a few days. So your marrow is just like pumping out those hematopoietic cells all the time. So when you plate these cells, you've got all of these stem and differentiated cells in the hematopoietic system. But they need very, very specific growth factors that you don't find just in serum, okay? So they die, okay? They don't get the growth factors. They want some IL-3 and TPO and EPO and GCSF, and they're not getting it out of serum, so they just croak, okay? So at the end, if you want to get hematopoietic colonies, you have to add very specific growth factors, very expensive growth factors to your medium. So if you just do this the way Friedenstein did it, you get colonies that are the stromal compartment, not the hematopoietic compartment. Okay, does that make sense? All right, and so he did this. It was really, really cool. I mean, you look back at his old papers, and they're, first of all, beautifully written because they wrote everything down then, including stuff that they didn't understand. You know, it's not like papers today where everything's just like written, like totally edited. And so what he found is that you could take those colonies, and he proposed that they arose from individual cells, and that you can take those, and now his methods are still used throughout the world, or variations on them, and study the basic biology. And you can also do in vivo assays of their behavior. So there's two kinds of assays that he did and that we still do. So I just want to show you, and this is not as you could see from those colonies I showed, there's a lot of variation in culture of how the colonies look, indicating that when you take these colonies probably came from cells that had very different differentiation potential or readiness to form tissue. So there's a lot of heterogeneity. It's not like there's these perfect stem cells and they all are clonal and give you exactly the same thing. So when you culture these cells, there's a tremendous amount of morphological variation when they grow out in culture. And this is just showing actually from one of my graduate students. These are primary cells. So you can see some are very spindle shaped and some are huge and spread out. And these are likely related to the differentiation properties. So given that there's this heterogeneity, how can we start to tease out the properties of these cells and think about how we would use them clinically? Okay, how they might contribute to disease, but how we might also use them. So this is a scanned-in picture out of one of Friedenstein's early papers, in which he did this kind of cool experiment. He took the marrow, or a bone fragment. He grew up the colonies, and he either mixed colonies together or he took individual colonies and kept them separate. And this was his attempt to look, he presumed that a colony came from a single clonal cell. And we can argue about that assumption later. But he did experiments where he took a single colony, saying this has a single clone, and asked what the potential was. Okay, now how did he do this? He did a bunch of things in culture, but what he pioneered was this thing called a diffusion chamber. And back then they didn't have syngeneic animals and so on, so you couldn't transplant willy-nilly. You couldn't just take cells and put them into another animal because they hadn't really invented all of these immune manipulation. So what he did is he had a little chamber that had a membrane on it that would let nutrients go back and forth, and he would put the cells inside that diffusion chamber. And so then in vivo he would look at their potential to become different tissues. Okay, so he takes either mixed colonies or he takes an individual one, which he presumed came from a clonal cell, and then he puts them in the diffusion chambers, and then he puts them back into the animals, and he would observe them. And what he would find when he opened up these diffusion chambers, he would find all these kind of crazy things happening. He would find some of them, nothing happened. Okay, that wasn't crazy because that's what you expect. Nothing's going to happen. Okay, he'd find bone, he'd find cartilage, he'd find just fibroblast-like mess, and he would find bone and cartilage together. Okay, so he would find all of these different things based, from different experiments. So these are, you're not going to be able to read all of this. This is just a huge summary of his data, saying how long he passaged them, whether they were clonal or not. But he's giving the outcome where he's got BB, bone, bone, empty, empty, fibroblast, fibroblast, bone, bone, fibroblast, fibroblast, not determined, bone and cartilage. So he really, really documented and gave the very first early evidence that a single stromal cell could give rise to more than one tissue. Okay, so this was revolutionary. Now we think about it, mesenchymal stem cells, they make everything. Everybody knows that. But back then, this was a hugely revolutionary experiment to see that what looked like a single cell-derived colony could actually make bone and cartilage from the same cell. It was very profound. Okay, so this was all done with animals, and then he subsequently started working with some other folks and showed that you could do this same procedure with human marrow, and that human marrow would do the same thing. And so I'm just showing here a standard colony assay, as most people will do it, where you seed what's the buffy coat, that's all the nucleate cells, so you get rid of the red cells if you can. You take the medium off and then let it go for a few days, typically about a week or up to 9 days, and then you can count the colonies. Okay, and different people have different thresholds for what they call a colony. This is just showing a picture. They're not hematopoietic. They're CD34, CD45 negative. CD105 is TGF-beta receptor endoglin, CD146. So there are some markers that have been associated with mesenchymal stem cell compartment. Okay, so this is a typical colony assay, and this is what is used to derive what people call in the literature mesenchymal stem cells. Okay, so we want to, in a minute, explore more about that. And so you can take these and you can trypsinize them, replate them, and expand them. And so that's what people use in the literature. So there are a number of sources of these. You can buy them commercially from a company called Lonza. There's an NIH center that Darwin Prokop runs in UT Southwestern, Texas A&M Health Center, and he provides them to the community at minimal cost. So there's a number of places. They all get them in a similar way of taking the nucleate cells, plating them, growing them up, and then characterizing them and freezing them down. So this is a standard assay. Okay, now, one point of debate, and we'll come back to this in a minute, is that when you try to determine the prevalence of colonies, it can get to be tricky. So this is George Muschler, my collaborator, has been very, very carefully trying to document the prevalence of connective tissue, mesenchymal stem cells. So I'm actually going to give you a little bit of mesenchymal stem cells became popularized by somebody I'll talk about in a minute. But it's, to me, a misleading term, because actually in those colonies, very few, in fact, in some cases maybe none, of the cells are actually true stem cells. What's the definition of a stem cell? What are some properties of stem cells? You guys surely know that by now. What's at least one property of a stem cell? Anybody? There's several. We just shout out one. What's one property of stem cells? Pardon? Okay, they can differentiate into every tissue in the body. That's actually maybe a little too broad. You're headed in the right direction, but they're pluripotent. They can make more than one tissue. Okay, so can we compromise on that? That there's more than one tissue that they can make. So in the case of mesenchymal stem cells, you would say, gee, at least bone and cartilage, and maybe bone, cartilage, and fat. Okay, so we would say, let's, and that's actually now commonly accepted that a mesenchymal stem cell can make bone, cartilage, and fat. Okay, but every tissue in the body is a little bit broader. Actually, it is broader than what the stem cell community would say. So it does have to make multiple tissues, multipotent, okay, or pluripotent, multipotent. What are some other properties? What's another property? Can you self-renew? Exactly. So it has to self-renew, and it has to be able to make at least a couple of tissues. So at the very least, the cells have to do those two things. Okay. And so when you plate these cells, it can be very hard. So there's a lot of interest in trying to relate how many actual stem cells a patient has in their marrow to some clinical outcome or some clinical phenotype. So George Mushler, for example, has been trying to plate marrow from all his patients and say, do patients with osteoarthritis have fewer or more stem cells? Do women, postmenopausal, smokers, you name it, category of patients, trying to understand how the prevalence of stem cells may vary with certain facets of the patient's disease state or other factors. But it can be complicated, and they've developed this automated colony counting because you can see the colonies can kind of run together. And so there's an issue of how to interpret some of the data. And so when you go out and read the literature, different labs have different ways that they judge these things, so you just need to be aware. But I want to come back to this issue of clonality and what it really means to be a stem cell, and how do we know that marrow really has stem cells. Certainly Friedenstein's original experiments where he did the diffusion chambers were pretty convincing, but they weren't quite all there because he didn't prove that it was a single cell doing these things. And it was really not until the late 1990s when Arnie Kaplan and a group from a company that got founded to commercialize some of these findings, Osiris Therapeutics, published a paper in Science really carefully examining the potential of these cells that the data were out there. So this is a paper, if you work with mesenchymal stem cells, you should be familiar with, by Pittenger et al. And what they did in this paper is they made colonies the standard way, and they went in and said, gee, we characterized these colonies, and you can go back and read the paper. They did all of the classic markers that people thought were for mesenchymal stem cells, and they did a whole bunch of in vitro differentiation assays. And so they took the colonies and they showed in vitro, they put them in media that would either, as because you've been doing this over the past week, you add different cocktails of things. So you can add certain things to the medium and the cells will become adipogenic. Okay, so these are just different donors. You can see an oil red O stain. If you take the cells and pellet them down in 3D culture and add certain things, they become chondrogenic. They make cartilage. And if you add different things, they become osteogenic, make alkaline phosphatase and deposit a mineralized matrix. So you could drive the cells that way. You take fibroblasts from the skin and do all the same things, and you don't see differentiation. So clearly these had potential for these three things. Let me see if I have, but I don't have. So what did they do in this paper that was so important? Because are you convinced if they took a colony and they did these differentiation, what would be another explanation than, it's a stem cell? What could be another, so if you took the colony that you got from the patient, so you take, if you take what they tried to say, actually what they tried to say here is they took it, they expanded it, I think 13 times, and they got the cells to differentiate. So they're saying, yes, they must be stem cells, because look how much we expanded them, and they're still making all these tissues. But what could be another explanation than a stem cell?
The cell itself is, the cells is the tissue that you are culturing.
Yeah, so if you have a mixed population, if you had a population of millions of cells, there could be some cells that make cartilage and some cells that make bone and some cells, and that was a huge criticism in the whole field of all of that work, all of this work. So what they did is went in and they took the cells that had already expanded, so many times, and they cloned out individual cells. So they now had taken those colonies, expanded them, they had, millions and millions and millions of cells, and they cloned individual cells out of that huge population, okay? And they took those clones and expanded them enough so they could work with them, and then they showed that individual clones could undergo multipotent differentiation. Okay, so that was really what convinced the field that there are, after you expand these cells, still stem cells present. Now, the efficiency was not very high, and to this day, the efficiency is not very high. And that's a huge topic of discussion or intense research that Roger Cam's lab and others have in the Singapore program and elsewhere. But this paper was seminal in saying, yes, there are at least a few stem cells still there after you do this. So it was a very convincing paper. And just a second. Okay, yeah, and so now there has been, this sparked, of course, interest when you went in and looked with markers and histologically, morphology. It's been proposed that there are these mesenchymal stem cells in all postnatal tissues. Most tissues have some kind of fibroblast-like cell, right? And often epithelial cells. So if you think about skin, you've got dermal fibroblasts and skin. So you certainly have heard that there are alternative sources. There are adipose-derived stem cells where you get liposuction. You can isolate stem cells that have many of the same characteristics as the one from bone marrow. So there's now tremendous interest in what the similarities and differences are from these so-called mesenchymal stem cells that come from different tissues. And I think it's very challenging if you're just coming into the field to go into the literature, because in vitro differentiation assays often can show remarkable things, but whether they will have the potential to actually carry out those functions in a sustained way in a real in vivo situation like in a person, there's not been an example yet where that's been successful. A couple of comments about where we find mesenchymal stem cells in the postnatal adult. They are typically found, so there's a lot of still controversy about where these cells reside in tissues in the postnatal adult. And they arise from the mesoderm developmentally, except actually there are some there's a little developmental background. The bones in your head come from the neural crest. So bones in the rest of your body and your jaw come from the mesoderm, but bones in your head actually are neural crest derived. So there's actually debate about whether you can form cranial bones from bone marrow that you get from the rest of your body, because your cranium doesn't have marrow. So a little bit of, just if you go into this, there's a little bit of debate and still people not sure. But there's a lot of evidence that's been presented that these cells are perivascular. And in fact, there was this very highly cited paper by Paula Bianco and Pam Roby that appeared in Cell a few years ago, indicating that CD146 positive cells that were perivascular were the mesenchymal stem cell. And they did a very complicated phenotypic assay where they took the cells, and human cells, and then they put them onto little ceramic carriers and implanted them in immunodeficient mice, and said, gee, it's only cells from bone marrow, these perivascular cells from bone marrow, that are capable of reforming a hematopoietic microenvironment. If you take perivascular cells from these other tissues, you can get bone, but it doesn't support hematopoiesis. So there still is a lot of debate about how tissue-specific these cells are. You can see some things, but not others. But what has come out since then, which is very interesting, is these authors made a big they really believe that CD146 is a particular marker of these mesenchymal stem cells. But since then, it's emerged some very interesting data published about a year ago that you can go into the marrow, and there's actually other markers that other labs have reported, like CD271, which is nerve growth factor receptor, that also seem to mark this mesenchymal stem cell compartment. And when you go in and you take cells, say with CD271, they do all the same things that mesenchymal stem cells do, except some of them don't express that CD146 receptor very much. They're CD146 low. But in fact, if you take cells that have this marker, but variable expression of the other marker, they have equivalent ability to form bone and cartilage in culture, and fat in culture. Okay, so this paper was showing that it doesn't matter about the CD146 expression; they'll all form these tissues. So CD146, as suggested by that earlier paper, is not the definitive marker, but in fact, what was really interesting in this particular paper is showing, again, these two markers. This is the one that was canonical, and this is another one that most of the community believes. What they showed, and I'm not going to go through this in gory detail, but they showed that this marker is distributed throughout the marrow. Some of the cells that express it are close to blood vessels, and some are far away. Whereas with CD146, the cells are all close to blood vessels and perivascular. So it's sort of like the looking for your keys under the lamppost thing, where they found that the CD146 positive cells could do all these things, and that stem cells are perivascular. But in fact, now later, you go in and find that they're positive. And so what they found, in fact, then, by going and culturing cells, they said, aha, the cells that don't have CD146 are pretty far away from the blood vessel. Maybe in low oxygen, cells just don't express CD146. Maybe if we culture cells in low oxygen, they'll still express CD271 because they have that everywhere, but in low oxygen, they won't express CD146. And that's exactly what they found. So these are FACS data where they culture in 21% oxygen, and you can, let's see, 21%, okay. Okay, so this is in so in 21% oxygen, you can see all the cells are pretty much expressing CD146, but in low oxygen, you lose the expression. Then you culture them in oxygen, all the sets, and they, again, start to express CD146. So it seems like this marker, which was viewed as the marker for a while of mesenchymal stem cells, is in fact regulated by oxygen, and there's more, there's actually evidence that that these stem cells are present all throughout the marrow. So the debate continues. These experiments are clearly very difficult to do, in part because you need, in the end, phenotypic assays that let you capture it. Okay, so I'm going to come back. Are there any questions about the basics? I'm kind of going through a lot of stem cell stuff, practical stuff. So I want to come back now to some of the ways that we're thinking about using stem cells in the clinic. And I already told you this. So what George Mushler has been doing is saying, well, maybe you don't need to take out bone from the iliac crest. Maybe you can just aspirate marrow, which is a much simpler procedure. The patients are going to recover from that very quickly. And so he developed a way to stick a trocar into the iliac crest and just suck some marrow out. Okay, and he's got it all mapped out to where you stick the needle in, and you can move it around, and so on and so forth. And he actually did this really important thing. And I don't know, is anybody here a surgeon? Okay, good. Anybody work with surgeons? No? Okay. So you can imagine, I know you work with surgeons sometimes, and they, they're very enthusiastic. They can fix everything with their, saws and hammers and stuff. And so one of, the kind of surgical mentality is, okay, if I take a milliliter of marrow, that's going to be good. But if I take 10 milliliters out, that's got to be better, right? Okay. That's just the way, surgeons are going to think. And so George actually went in. He's a very careful person. And he went in and he said, how does the actual prevalence of these stem cells, or connective tissue progenitors is what we prefer to call them, how does their prevalence in the biopsy or aspirate vary with the amount you take out at one time? Okay, so how does the concentration of them vary? So he did a series of experiments, and where he took 1 mil, 2 mils, 3 mils, 4 mils, and he found that the number of cells per nucleated cells per sample decreased with increasing volume. Of course, because as you aspirate more, you ultimately just start sucking out peripheral blood. So first you get the marrow, and then you just are sucking out blood. And that doesn't have, it has some stem cells, but hardly any. So you eventually go down to some baseline that's the prevalence of cells in peripheral blood. Okay, so his preferred aspiration volume is somewhere between 1 and 2 milliliters in order to get the stem cells. But it takes a lot to educate surgeons that it's better to do 10 aspirates of 1 mil each than 1 aspirate of 10 mils. So what George has been doing is actually taking this into the clinic. And what he reasoned is that you could take the marrow aspirate, as shown here, which has the stem and progenitor cells in it, and pass it over a matrix that would capture the cells, and all the cells, but maybe you could select for the stem and progenitor cells, and then that would be sort of your surrogate autograft. Instead of autograft, you now have the marrow, and you put it on a scaffold and put it in. And so together, actually, I co-invented a process called three-dimensional printing for making tissue regeneration scaffolds, and it's been commercialized by a company called Therix, which is now part of Integra, and they have have a range of clinical products for bone regeneration that are 3D printed with very precise microarchitectures out of, in this case, beta tricalcium phosphate. So these are some so-called bone void fillers that actually George designed their shape. So 3D printing lets you make a very specific shape, but also a very specific microarchitecture. So when you pass marrow, you can just see the marrow here. You can count how many, you can take a little sample of the marrow that goes in and of what comes out, and assay for the prevalence of those progenitor cells. And it turns out, for reasons that we don't really understand, the marrow, passing it over the scaffold actually selects for and concentrates the stem cells by about a factor of 4. And it could be that their size, that they just kind of get caught because they're small and they get caught. We're still not sure, and it happens in lots of different kinds of scaffolds. So what he's done then is go on, and others as well, and show that by using this so-called selective cell retention process, you can concentrate the marrow, you can actually improve bone healing outcomes. And so it's been verified by other labs. So he's using this in the clinic, and a barrier to getting it more widely adopted turns out to be how the FDA is going to regulate this process. So they're going through a very, it seems simple just to do this, but then the FDA wants to regulate it and do a controlled clinical trial. So the thing that we've been concerned about together on the molecular side is improving this process of adding marrow to scaffolds, particularly because when you go in a large defect, it's much harder to heal a big old defect in a sick person than it is to heal a little defect in a rabbit or a mouse. And one of the challenges, as I've alluded to several times, is that the human marrow has, there's a huge variability in what individual patients have as prevalence of stem cells. So this is the same number of nucleated cells plated for 5 different people. And you can see this one has very few, and that one has a lot of connective tissue progenitors. So this patient, if you put marrow in a graft, this patient may not get better because maybe they just don't have enough stem cells. So we've been very interested in whether we can increase the molecular environment for these stem cells by parsing what will happen to the cells as they're transplanted. And so initially they have to survive, then we want to have them proliferate, have blood vessels grow in, et cetera. So we are thinking about the whole life cycle of these cells as they go into this very hostile environment of that wound. And in thinking about that, there's certainly, you probably have read about bone morphogenetic proteins, which are in the clinic for bone healing. You add BMPs, which are very powerful differentiation factors for bone, and they can stimulate healing. It's a product by Medtronic. Sometimes used off-label. It's actually killed some people using off-label. But those tend to act a little bit downstream. We're interested in these huge defects where there's a deficiency of stem cells, so we may need to act further upstream than the BMPs act. And so we know from the literature that the epidermal growth factor receptor is important, is essential actually in both development of bone prenatally, embryologically, and in maintenance of the stem cell compartment in bone postnatally. So there is, if you take bone marrow out and plate it to get those colonies as I described, pretty much everybody plates in serum. Okay, but there's been a couple of studies of how could you derive colonies in serum-free medium. It turns out if you plate on fibronectin and you add EGF, you can derive the colonies. So EGF alone is sufficient to get the colonies. So it's a very important growth factor for early progenitor function. So we've been coming back to that EGF question. We've been asking, can we take this growth factor, and I've already gone through these, and actually use it in a regenerative way for bone healing, thinking about going to the clinic. And a reason that George, the other option for how you might treat these patients is to take their marrow aspirate and culture expand it, expand those cells out and put them back into the patient. Anybody want to offer some pluses and minuses of that approach? So first of all, taking something out of the OR is going to be expensive. Doing that is expensive. Number two, we really haven't learned how to expand cells and keep them in a primitive state yet very well. And in fact, the company, Osiris Therapeutics, that Pittenger et al. 1999 science paper, it was a company founded to do exactly that, to expand the cells, put them back in the patient to heal bone. And they ended up failing as a business model because it was too expensive and it didn't work very well. So there's a limit right now. Maybe that'll work, but contrast that to treating the patient in the operating room. Okay, so you aspirate their marrow, you do a procedure that takes 10 minutes in the operating room, and you put it right back in, and you send the patient on their way. So it has the potential to be much lower cost, also safer because you're not potentially contaminating the cells or getting them, So there's a lot of advantages if you can to do it in the OR. Of course, it has to be effective. So that's why we've been focusing on this. Now, what's interesting, if EGF, first growth factor ever discovered, there are two commercial products, okay? I'm showing you one of them. Okay, so it's not for tissue regeneration. This is, it turns out if you inject EGF as a soluble molecule into sheep, it circulates with a half-life of about 2 days. And it turns out EGF receptor is very important in hair growth, and it regulates the stem cell compartment in hair follicles. So if you give a huge amount of EGF, it downregulates the receptors, and the hair doesn't grow properly. It grows very thin. And a couple of weeks later, when the hair reaches the surface, it's very thin and brittle. So with sheep, that lets you just pull the wool off of the sheep. And this is really important because in Australia, kids don't like to stay on the farm. Sheep shearing is actually a skill that's hard to learn. So you can use this BioClip to shear sheep. And that's a commercial product. The other commercial product, if you go on eBay and type in EGF, you can find like these $200 little vials of anti-aging cream that have EGF in them. Seriously. And it says, the FDA doesn't, approve that this is, effective, but I'm 95 years old and I use those anti-aging creams. Okay. So one of the problems then in delivering EGF is that if it's in a soluble form, cells internalize it, so the life cycle binds to the receptor, this dimerization signaling starts, but then you get an internalization process and downregulation of the receptor. Everything gets degraded. So what we've been working on is a way to tether this to the surface, and remember, dimerization is involved, so we developed some polymer methods to cluster little EGF ligands on the surface. So here's a tether, here's the EGF. What I'm showing here is a crystal structure of dimerized EGF receptor showing that we can tether it in a way that will allow dimers to form. Okay, so we have gone on now, and we're starting to develop this for clinical application and shown that soluble and tethered EGF signal in a completely different way. Well, not completely different, but you certainly get different phenotypes in that this is with some human cell lines that you no EGF, with EGF, and with tethered EGF in terms of how you can see these cells are spreading much more effectively than these cells. So the soluble and tethered are signaling differently. And you can enhance colony formation on a bunch of sort of model substrate, adhesion substrates by tethered EGF, but not by soluble EGF. And so we've translated this into a way to put tethered EGF on those tricalcium phosphate kind of scaffolds that are clinically used, and together with George Mushler have shown that you can enhance the colony formation from human marrow by putting EGF in a tethered format onto the surface. So this is a control, and this is with tethered EGF. This is actually a pretty hefty enhancement of colony formation. When you've been doing these experiments for a long time, it's about a 40% increase in the number of colonies you get. So this is now, we're moving into animal studies. We actually just got yesterday some of our first data that looks very promising. And so we'll see, it takes a long time to get these in. The final thing, so that's the colony formation. Remember, when we go into these grafts, it's a horrible environment. It's very hostile. There's no no oxygen, there's all these inflammatory cytokines. So a very fascinating thing, and I apologize, the graph is taken from here, is that tethered EGF acts completely differently in terms of protecting cells against that environment than soluble EGF. And so if you have human mesenchymal stem cells, connective tissue, those culture expanded cells, here they are growing on the surface. If we give a pro-death cytokine that would be present in the wound environment, you can kill them. You can see them all killed here. And I don't have the graph, but this is just showing there's a lot of cell death. But if you now have them cultured on tethered EGF, they're protected. And in fact, if you add soluble EGF, they're more dead than they are here. So you actually potentiate death with the soluble, but you protect them with the tethered. So this is one of the reasons we're going in vivo with it, because we feel like we might be able to protect those early progenitors during this initial inflammatory milieu and take them on. Yeah.
So do you know how much in body, how much of the EGF is soluble, like, versus?
Most of the EGF, so it depends on where you are. So EGF and TGF-alpha soluble are very important in the gastrointestinal tract and in some other barrier epithelia. In places like bone, most of the ligands are not EGF. They are, in fact, amphoregulin or HBEGF, or things that bind to matrix or maybe acting in a juxtacrine way. So in fact, our putting EGF tethered was a way to mimic the matrix-matricine delivery. It's just that EGF itself as a molecule is really nice to work with compared to some of the other ligands that are biochemically, like HBEGF, heparin binding EGF, is the most positively charged protein that we know of. So it's a very difficult molecule biochemically to work with. So we use this little one that binds to the receptor. But most of them in the body are normally somehow embedded in matrix, bound to matrix, or still sitting on the cell surface, and it's only a little bit that gets released proteolytically, except in special places like the GI tract where you have it bathed there, and in milk. So in nursing, because the baby needs EGF receptor stimulation in the GI tract, so there's a lot of EGF receptor ligands in milk. Yeah?
Do you know, does EGF that binds to collagen, does it remain functional?
EGF doesn't have a high propensity to bind to collagen per se.
We've seen quite a bit of binding to collagen type 1.
Okay, it may depend on the format. It's not reported in the literature to bind to collagen, so it may be a non-specific kind of, but we also have seen that, like we make a collagen gel, lots of stuff like a cytokines, just there's a lot of non-specific absorption because they're low concentration and they'll bind, and if we block with albumin, we can inhibit the binding of EGF to collagen. So I'm not sure it's a specific, I think it's a non-specific binding because in Ben Cosgrove's paper, we did some experiments like that and found that TGF-alpha, which is very similar to EGF, also would bind, but we added albumin to just kind of block non-specific binding, and it didn't bind.
Okay, fair amount of binding.
Yeah, I think collagen gels or any kind of gel, there's going to be a lot of non-specific binding, and separating what's non-specific from specific can be important. And you may want to block non-specific binding depending on what your experiment is. But I would think that, so I don't know the answer to that because I haven't investigated it. But I believe, I can believe it happens because we saw the same thing with collagen gels and just blocked with massive doses of albumin to inhibit that, and actually did ELISAs and showed that you could protect it. And I think the data are in Ben Cosgrove's paper. So I just want to show this one last slide because I just think it's very interesting. So I mentioned that other tissues are sources of mesenchymal stem cells. And these are actually derived from menstrual blood. So you can collect menstrual blood, and it has cells. The fibroblasts in the endometrium have almost identical properties to mesenchymal stem cells. They have CD146, all the same markers. In this paper, they were investigating the properties of the cells shown in A and trying to show they could differentiate in cardiomyocytes and actually function in vivo, the same way that you would get with, people have done with bone marrow-derived cells. So it's hard to know whether, these things would ever play out in the clinic. Other people have taken menstrual blood-derived mesenchymal stem cells and used them in animal models of Parkinson's and shown effect. So there's a lot of exciting things. How much of this will play out to be an enduring phenotype that will be operative in a human patient is still not clear, but there's certainly a lot of stuff going on to investigate these things.
Now, I know you have something else to go to, but do you have time for some questions?
Just a couple of quick questions. Yeah, I have 30 people waiting in another room for me because there was a scheduling conflict for a meeting that starts at 9, which started at 9, a meeting that I'm running.
The difference between the cellular one and the cellular one, is it mostly because the cells now have another place to grab on, or?
The number of molecules is very small. The number of EGF molecules is very, very small. Mesenchymal stem cells only have a few thousand EGF receptors. It's likely the same. I mean, in fact, EGF itself is anti-adhesive, so you can inhibit cells adhering. It's very complicated signaling, and it's almost surely not because there's only a very few molecules actually there. So it's more the signaling mechanisms that are inducing it.
Thank you very much, Linda.
Okay.
We will take